Okay, hello everyone again, and welcome to the final talk of the session. Professor Goss earned her bachelor's and PhD at the University of Durham and performed postdoctoral research in the biochemistry department at Cambridge. After holding positions at the University of Exeter and the University of East Anglia, she moved to her current position at St. Andrews, where she is a professor of biomolecular and organic chemistry. Her present research focuses on elucidating and engineering the biosynthesis of natural products at the chemical and genetic level, and in blending synthetic biology and synthetic chemistry to make new-to-nature natural products. And this morning, she'll be presenting GenoChemetics, blending synthetic biology with synthetic chemistry for molecule making. And the virtual stage is yours, Rebecca.
Thanks so much, Ian. And thank you to Nigel for this really kind invitation to what's been such an exciting symposium and such a really fantastic morning of events so far. So as Ian says, I'm going to be talking about GenoChemetics, and I'm really going to be talking about blending that full palette of synthetic biology with synthetic chemistry. And I'm dedicating this particular talk to my father, Christopher Goss, who sadly passed away on Friday night. But this is a very broad brushstroke presentation that I'm giving, very much looking at the opportunities, looking at the landscape. I'm going to start with a research snapshot. Then I'm going to move on to discussion of GenoChemetics, a fusion that we're working on within my team. And I'm going to look at finding new tools and developing new tools to add to this palette, if you like, new tools for GenoChemetics. We've just had a review published in ChemSoc Review. It's just come out a couple of days ago, very much with this theme of the chemistry, synthetic chemistry, and the synthetic biology complementing each other to enable molecule functionalization. So starting with that research snapshot.
So just a heads up, Rebecca, I'm not sharing your slides right now.
Am I not? That's very interesting. Thank you. Am I screen sharing?
I don't think you are.
Right, let's try again.
Perfect.
Okay,
you just need to go into presenter mode.
Great.
Great, you're awesome.
Okay, so thanks, Ian. So hopefully you guys now have my first slide in presenter mode. And as I was saying, we're looking here. I want to give you paint you a really broad brushstrokes picture of the opportunities of blending together synthetic biology components and also synthetic chemistry components, extending those pathways. And as I said before, I'm dedicating this lecture to my father, Christopher Goss. So very quickly, I am sweltering at the moment in Scotland. So St Andrews is based here, just a little bit north of Edinburgh. It's an absolutely stunning place to come and visit. So please do feel free to strike up a conversation with me if you fancy collaborating, or if you just fancy a visit to St Andrews School of Biology or School of Chemistry. We have a very nice new refurbishment going on. We'll be opening brand new labs in this stretch here in May of next year. And we have some really nice labs within this biomolecular sciences facility, which really brings together scientists working in molecular biology, but coming from medicine and computational science, from biology, from engineering. And St Andrews is a beautiful, scenic place. The university is 600 years old, but as you can see, the labs aren't 600 years old. And here we've got a picture of the ruins of St Andrews Cathedral, the castle, and some attractive to some greens where a lot of golf is played, with St Andrews, of course, being considered one of the homes of golf. So I said I was going to start with very quick research snapshot. So my research team and I are passionate about natural products in the very broadest sense. We're interested in discovery, looking at ecologically unusual niches. We like using either structure-guided approaches for discovery, bioactivity-guided routes to discovery, and of course, omic routes to discovery. And we base a lot of our work now on looking for interesting motifs within gene sequences. So moving on from this top left quadrant, this takes us into a heartland for my research group of biosynthetic elucidation. We're particularly drawn to molecules that have interesting motifs, chemical motifs in them, and interested in understanding how the enzymology works, how the chemistry works, and the general construction of these motifs. So for example, here is an antibiotic that we work on called pasidomycin. It's one of the uridyl peptide antibiotics. And although at first sight, it might look rather bland with its peptide backbone fused to this nucleic acid here, if you look a little bit more closely, there are some really curious features within it. So for example, rather than standard tyrosine, there's metatyrosine. And we've been looking at the enzyme involved in the production of this. The N-terminus of the peptide here and the C-terminus here, yes, but there's a flip in the sense of that peptide, which we believe protects the molecule from proteolytic degradation. So this diamino acid, diaminobutyric acid at the center, reflipped by the installation of this carbonyl. And perhaps the most curious feature of all within pasidomycin is this modified nucleic acid, where this 3' hydroxyl is missing. And we're working on the enzymes there and interested in harnessing them as potential ways for being able to access new antiviral agents. So that understanding of biosynthetic pathways, that really leads us on to another passion within our group, which is developing enzymes as tools for synthesis. Either individual enzymes or full pathways, harnessing a pathway to be able to very quickly get access to the natural product, but also to refactor it and to be able to access quickly new to nature natural products, either through modification of the biosynthetic gene cluster or by introducing additional chemical steps in concert with those pathways. Okay, so natural products. Many of this community are really, really interested in secondary metabolites, and it's great to see that there are separate streams focused in on the secondary metabolites here. But natural products, why should they really be so important in 2021? Well, these molecules remain privileged structures. Made by biomolecules, they are naturally predisposed to interact with biological targets. And so perhaps it's not so surprising that natural products form the basis for over 80% of current antibiotics in the clinic and over 60% of anti-cancer agents. So they're highly medicinally relevant, but if we're to really to understand how they interact with the target, or if we're to take them from their natural functions and to improve them for human medicine or for use in agriculture, then being able to generate analogs is really, really important. And then we can see which components on these molecules are super important, and we can start to tune bioactivities, bioavailabilities. But one of the criticisms that is often levied at natural products, secondary metabolites, is whilst they're beautiful, they are very, very complex, and it's very difficult to systematically make changes. This actually is a misperception. Misperception based on the complexity, the architectures of these molecules, and the difficulty using conventional chemistry of just changing, let's say, a methyl or a hydroxyl. But of course, this community as a whole can play the power of true synthetic biology and enzymology as well as synthetic chemistry. Okay, so let's look back at that misperception and the standard ways in which analogues of natural products were made previously. So, of course, there's a route called semi-synthesis, where a chemically reactive, chemically orthogonal component on the molecule is further functionalized. But you're really, really straitjacketed by the native chemical orthogonality in that molecule. Wouldn't it be nice to add in a truly orthogonal and reactive handle into positions in these molecules? The other way of making natural product analogues has been that of total synthesis, which is a really valuable approach for making analogues of natural products and the parent compounds themselves. But it's really hard work, and it's often paralleled to mountain climbing, being not only tough, but long-winded and sometimes dangerous. But of course, here as a community, we can start levering true natural routes to be able to access analogues of these natural products. So harnessing those microbes, harnessing those enzymes, harnessing those cyanobacteria to be able to deliver differences in those molecules and be able to very rapidly make new natural products. So a really old approach to doing this, it's been around about 60 years now, is precursor-directed biosynthesis. So here on the right, we have a precursor of rapamycin, or a variant of rapamycin. And the total synthesis of rapamycin, the first total synthesis of it, took 102 separate steps and a total of about 50 postdoc years at least. Whereas this microbe in the middle, the superstar Streptomyces hygroscopicus, can make it in 3 days. And if you make a mutant which is deficient in the enzymes that are able to make the natural starter unit, you can feed in a whole series of either Aldrich catalog building blocks or synthesize analogues of these yourself and then feed them in. So this looks like a really small, little innocent molecule, but in fact, this fluorinated alternative to this starter unit, each analog took 6 to 7 steps to be able to make. So it's clearly not a trivial endeavor. And then the microbe makes it look easy by being able to incorporate it and make, in many of the cases with these fluorinated analogs, up to 100 mg per liter of that new fluorinated material in just 3 days. So still very challenging. So the concept with GenoChemetics is, let's inject some true chemical orthogonality into the natural product first. So let's find a gene encoding an enzyme that will install a truly chemically orthogonal, a truly reactive handle into that natural product, acting in concert with the existing biosynthetic pathway, so that we just have that one handle there. There's no need for any further protection around the natural product, but we can point to point affect change at the point where we've installed that new handle. So that's the concept behind GenoChemetics. Install a gene that puts a chemically reactive orthogonal handle onto the molecule, and then that functionalized natural product can be further diversified for structure-activity relationship studies, or perhaps it could be fluorescent tagged, maybe in the presence of a living organism, or Perhaps it could be utilized to aid purification of the natural product or to present it in one particular orientation so that its true interactions with various different potential targets can be explored. Okay, so that's the concept, to be able to take any natural product and to be able to march round not just one handle, but series of different handles with different chemical reactivities. And these handles should be fairly rare. They shouldn't be regularly found within nature, so that you have that true discernment of the chemistry, if you like, just acting on those activated species. Okay, so that's the big picture, the big concept with this. Obviously, there are a lot of challenges to deal with on both the genetics side, molecular biology side, synthetic biology side, as well as the chemistry optimization. And I'm focusing here just on the utilization of aryl halides. And I'll very much gloss over the synthetic chemistry that we use. But if anyone wants to talk to me about that in more detail, I'm very, very happy to pick up conversations by email and future teleconference conversations. Okay, so why are we alighting on the aryl halides? So halogenation is shown to quite often improve bioavailability. Just what looks like a fairly innocent change of a carbon-hydrogen bond to a carbon-chlorine or carbon-bromine or carbon-fluorine bond can massively increase the lipophilicity of that natural product. It can also have a good impact on bioactivity. But most importantly here for the GenoChemetic approach, it enables cross-coupling. So a reaction where a carbon-halogen bond can be diversified by a series of different reactions. So the big picture, let's take a gene encoding a halogenase, let's get it to play alongside the pseudomycin biosynthetic pathway. Let's see if we can get it to install a halogen into this molecule. And then let's see if we can develop some chemistries to very mildly, preferably in the presence of the living organism, be able to further modify that carbon-halogen bond. So when we started this out, surprisingly, there weren't any carbon-carbon cross-coupling modifications of free, unprotected halotryptophans. And this was something that we had to very much build from scratch. This strain hadn't been transformed previously. We had to develop protocols and suitable plasmids for that as well. So excellent postdoc in my group, Dr. Sabine Gruszczo, took pRNA. pRNA is responsible for the very first step in pyrrolnitrin biosynthesis. It's an enzyme that was worked on by Karl-Heinz van Pay. And it CH activates, or it installs a chlorine at the 7 position of the indole ring of tryptophan. Then there are subsequent enzymes and steps that take this through to the full natural product, pyrrolnitrin. So taking pRNA, could we use it to work alongside the pseudomycin biosynthetic pathway? So we took our pseudomycin producer, Streptomyces coerulirubidis, and we worked through a large series of different plasmid constructs with inducible promoters, constitutive promoters, integrative, non-integrative plasmids. And we were eventually able to find a system that worked well for the transformation of this strain, and where we could produce up to around 70 milligrams of our chloropseudomycin. Next, to bring in the chemistry, which I'll scoot over pretty quickly, but we were able to develop conditions that would work on a crude extract or on the purified natural product. And using Suzuki-Miura chemistry, we could change through a variety of functional groups, and some of them are shown here. You probably have noticed, though, that the temperature's rather high, and we're doing this in a microwave. And I said to you before about my big picture vision being that it would be great to truly hyphenate, truly extend a biosynthetic pathway with synthetic steps. So we've either got to do this in a thermophilic organism, or we've got to bring down the temperature of this chemistry. So carbon-chlorine bonds are short and low energy. If we extend out to a carbon-bromine bond, we can start to perhaps bring those temperatures down. So let's make our bug make the brominated analogues. So we took a series of bromide salts into this, and what we found was Streptomyces caerulea rubidis was very, very unhappy with any form of bromide. It was very sluggish in its growth. Mostly it died. And very strangely, we saw downregulation of most of the biosynthetic capability of the strain in the presence of the bromide salts. So what we did was we screened a series of other potential host strain chassis strains for their ability to tolerate different halides. And fortunately, Streptomyces silicolor, a very well-worked strain, the gold standard Streptomyces host, was able to tolerate good levels of the various different halide salts. So we pulled down, we found the biosynthetic pathway from Streptomyces caerulea rubidis, and we heterologously expressed this in Streptomyces silicolor. We introduced the additional enzymes that are required for the introduction of the metatyrosin that I showed to you. We could either have the metatyrosin played, or in the absence of these genes, we could get to the alanine-containing variants. We introduced our halogenase, and we could get through to our brominated analogues. Then it was a case of really investigating what happened to the microorganism that was producing that brominated pasidomycin. What happened to it when we tried to introduce an organic chemistry step to it? And what we saw was that there was a level of, there were various different ligands that we couldn't use in the presence of the living system, but also there was an impact, of course, of the presence of this living organism and the compounds it was producing on this catalytic pathway. So, for example, the wonderful sugars, the wonderful amino acids that you're required to be able to ferment this strain or are being produced by the strain, the amino acids coordinate to the palladium species, stopping it from being a good catalyst. And so you can replace those instead with different sources. And the sugars coordinate to the boronic acids, taking them out of the reaction. But instead, we were able to replace that with glycerol. So it's very much an iterative process of looking at the impact of the chemistry on the organism and the organism on the chemistry and its metabolites on the chemistry. Okay, but we could play that and we could extend the pathway, and we could do these reactions either at 20 degrees or 27 degrees or 37 degrees. And we were able to generate our modified tryptophans or our modified pasidomycins. And depending on what our cross-coupling partner was and what our diversification was here, we saw evidence of product held within the cells, indicating that a level of the chemistry was perhaps going on inside the cells. So if we could take a step further and move toward iodination, that might be even better. So we've already enabled much lower temperatures for the chemistry by changing from a carbon-chlorine bond to a carbon bromo bond. If we went to the iodo system, this could be enabling of even lower temperatures. So we started to look for new halogenases that might be able to enable the iodination within these systems. And we took very much a genomics-informed approach based on what we knew about the active site of these enzymes, what we knew about the mechanism, and we started to mine through this data. So previously, when we started this work in around 2012, there was a very, very small portfolio of halogenases known. But going out into that deeper genomic space, we were able to significantly extend upon that. So highlighting in on VIRX1, so VIRX1, we chose as one of about 200 halogenases that we're studying because of its level of proximity to things like pRNA, which I've introduced you to before, but because it looked sufficiently different. And when we modeled the enzyme, it looked as though it might have a nice wide open active site. So our approach with this was for each and every halogenase, we'd produce the protein, then we'd screen 600 compound library by UPLC, and then we'd validate those real peaks on the UPLC by using LCMS and seeing whether or not we saw the halogen isotope patterns. So going from this very small, discrete handful of substrates, now for one wild-type enzyme, you're seeing with the wild-type a much broader series of substrates that can be brominated or iodinated. But one of the things that was really fascinating to us with this wildly different wild-type halogenase was perhaps its role. What is it really doing in nature? And we've seen some lovely talks on photosynthesis this morning, and there've been really great talks throughout on cyanobacteria. VIRX1 comes from a cyanobacteriophage. It infects Prochlorococcus, Synechococcus. So we're really, really curious as to what it might be doing, what its real role is in nature, able to activating molecules for that further synthetic functionalization. It's the natural world versus the laboratory world. So I'm privileged to work with a really awesome set of co-workers, either on the synthetic side, there's a true blend from the synthetic chemistry through to the synthetic micromolecular biology. We've had some particular stars on this project, the Saints at St. Andrews. So Danai, who found that last enzyme that I've just spoken to you about, Hannes, who worked with Danai. Hannes, a joint student between Jim Naismith and I, and worked on the structure of VIRX1, and then some real synthetic experts, just to name two, Sunil Sharma and Rose Lynch. Really grateful to our collaborators, really grateful to our funders, and I thank you so much for your kind attention and for bearing with me when I didn't manage to share my slides in those first moments.
All right, thank you very much, Rebecca. That was absolutely fantastic and really demonstrated the power of combining living systems with the engineering, the chemistry. I'd encourage anybody in the audience to drop any questions into the Q&A box, but I'm going to go ahead and kick things off with maybe more of a general question. I guess I'm guessing there's a lot of potential of taking advantage of biological systems to specifically alter or to define the chirality of functional groups of a molecule. In practice, is this a feasible approach?
Absolutely, and there are so many really cool strategies that you can use to do this. So on one hand, you can take advantage of these beautiful PKS systems that I very quickly scooted past to be able to set those stereochemistries on the positions of the hydroxyls, for example, or the positions of those methyl groups. So there's a lot that can be done there. On a single enzyme point of view, rather than an enzyme as a component of a multifunctional pathway, there's great opportunity to be able to use enzymes as a sift. So quite usually an enzyme will work on just one enantiomer, leaving the other untransformed. And as you can see with halogenation, or as I told you in terms of halogenation, the difference in lipophilicity is huge. So there's great opportunity there to be able to do deracemization and functionalization.
Fantastic, thank you. And another question appeared, but it disappeared again from the Q&A box. It's back again. Not sure what's going on there. Do you have a ballpark number of how many enzymes could be expressed or purified, and how many show activity in the genome-guided screens?
Gosh, sky's the limit. So so a ballpark for how you do it. It really, so in terms of being able to do the discovery, it's using informatics tools first. So the first informatic discovery is getting faster and faster. We like to validate using a number of different approaches. So we after alignments, we'll then do our structural modeling, etc. And the computational timeframes for these steps are reducing. And of course, there is huge opportunity now with AI to start to think about faster sifts with that. So that's a really exciting area. In terms of then taking them through from gene to express gene to produced soluble protein, you can be a little bit strategic on this. So if you set your search guide so that you're going for genes that should produce reasonably well-folded proteins, then you can stack the odds. And you can also use other stacks, such as your choice of tag, incorporating solubility tags, etc. How quickly can you do that? So starting with the best informed approach, I think, is really important, Ian. But then it's a question of whether you can easily multiplex, whether you have access to the robotics.
Yep, absolutely, yeah. All right, well, again, that was absolutely fantastic. And thank you very much, Rebecca.